why don't we move into, we'll have Dr. Nael Nadif Kasri join in. So Dr. Kasri is going to be presenting a discussion around the topic of leveraging stem cell technology to dissect the cell type specific contribution of EHMT1 in Kleefstra syndrome. So Dr. Kasri, I'll pass it to you.
Thank you, Jeff. I hope you can see my slides. Can you see my slides? Yeah, presentation?
We can.
Okay, perfect. Yeah, thank you very much. Well, first of all, for this putting this fantastic conference together. I think it's always an honor to be able to share our research, our plans with you, our strategies on how to work on Kleefstra syndrome. So my name is Nael Nadif Kasri. I'm a professor at the Radboud UMC, and I work together with Tjitske Kleefstra, Annette Schenk, Hans van Bokhoven, and also Joost on Kleefstra syndrome for many years already. And particularly my lab is trying to understand really what is happening now in those nerve cells of patients with Kleefstra syndrome. And that's actually a very difficult task, because essentially the human brain is inaccessible. So if we really want to understand what is different in a patient's brain, then one would like to be able to enter that brain to look at what is different. We want to manipulate it, we want to test some hypothesis, and so forth and so on. And it's obvious that this is not possible with nobody's brain, and so one has to find other ways to do that. And so for the many years, researchers have found surrogates for the human brain, basically by investigating animals' brains which have actually the same mutation as which we can find in patients. And this has been very helpful and is still very helpful. But obviously, a mouse brain, for example, is not the same as a human brain. And so what we set out for a few years ago already is to really trying to understand how the nerve cells of Kleefstra patients function, and how they potentially function differently than healthy controls. And so for that we make use of stem cells. And today I would just want to share with you our strategy, how we use them. And probably many of you have already heard about stem cells and how we can use them, but I really want to take you step by step along the way. So what we can do is actually we can take for any individual, we can take either blood or fibroblast or hair cells, and we now have a possibility to convert those cells back to stem cells. And basically stem cells are, you can see them, the cells that are at the origin of life. And so they have the capability of becoming any cell type that you want. And so having them in a dish in the lab allows us now to choose to which cell type to differentiate them. And obviously, as I mentioned, we are interested in brain cells, so actually in all the cells that are present in the brain. Those include nerve cells, but also non-neuronal cells. And so here you see an example of those beautiful nerve cells. I always show this because it always gets me excited. You see the complexity of those nerve cells. And this is an example of after 1 month in such a dish. You can start from the stem cells and you can get these very complex cells. And so in green, you see in principle all these dendrites, we call them. And on those dendrites, those cells receive information from other cells. And the spot where, the place where this information is arriving is actually at all those red spots that you can see along the green dendrites. So there's actually an invisible other nerve cells here that makes contact with this green cell here. And so this is how information is basically transmitted in the brain, but then not with few cells, but with billions of cells. And that's now we have the possibility to have this in addition, to study this in addition. So if we zoom out a little bit and we look at the bigger picture, what we actually want to do is being able to start from patients, generate these stem cells, which you call induced pluripotent stem cells. And then from those stem cells, we generate all types of brain cells. And I think today we'll hear more about it from other speakers, but I will really be focusing on brain cells themselves, on nerve cells. And once we have them in addition, obviously, we want to do something with it. We want to measure something. We want to be able to measure something so that we can say, oh, this is different than the healthy control. And I will show you the way we do this. And that's what we actually called phenotyping. And once we have a phenotype, we try to use this to understand how this phenotype comes about, but also we can use it, of course, to try to test all type of compounds that are available to see whether we can bring it back to the normal situation. And this, obviously, the last part is the most interesting, but also the hardest part. So how do we now measure? What do we measure from those nerve cells? So as I mentioned, we can generate those nerve cells now in dish. We can compare nerve cells between Kleefstra patients and controls. And something that is really important to mention is that now every nerve cell that we generate starting from a particular patient has exactly the same genetic information as the donor. So it means if a patient has a certain mutation in the EHMT1 gene, then that exact same mutation will be present in this nerve cell, in addition to all the other differences in the genetic background. And so this is what we do, but what can we then measure? Now, what we know is that those nerve cells need to communicate with each other. So they need to talk to each other. And so we know from many disorders, including Kleefstra Syndrome, that often that's actually the problem. It's the communication between the nerve cells that seems to be different. So we like to grow those nerve cells in what I call a smart dish. And that's a dish that has electrodes embedded in the dish. So this is not a real image, let's say. How it really looks like is what you can see here on the left. So you can see here and zoom in actually of the electrodes, which are indicated in this as this black dots, and the structure that you see here, this gray structure that you see here on the background, are actually the neurons that are the nerve cells that are growing on top of the electrodes. And so if we put it very schematically, we have this grid here of electrodes, and then we can grow the cells on top of the electrodes. Now, the nice thing is that, as I mentioned, the neurons, they like to communicate with each other. And so we can start to pick up this communication through those electrodes. And so that's exactly what we do. And so here on the bottom, you see a color map and the changes in color. And the more red you see, the stronger the communication is happening, and the bluish, then you have no communication. So we can measure directly actually how nerve cells from a particular patient or person, actually how active it is and how it communicates with each other. Now, obviously, we are scientists, and we want to be able to monitor that a little bit more quantitatively. And so what I show you is a typical example of how those measurements look like. So we call this a raster plot. And so every line that you see here is basically one of those electrodes. And if you go from left to right, you have time. And so every vertical line that you see now, those black stripes, is every time that a neuron actually communicates with another neuron, another nerve cells. And so once in a while, you see that they actually all communicate at exactly the same time. So this means that what we call the cells are in a network, and cells like to be in a network, just like people like to be in a network. And so the healthy population of nerve cells will actually always show this typical pattern. And analyzing this in detail really allows us to look to really ask the question, when are those nerve cells communicating, how often they are doing this, with who are they talking to, and also how loud are they talking. And so this gives us really a lot of very valuable information. And I really want to re-emphasizing this is really, we can do this now for every individual patient. Now, the nice thing is then if we, or the most, I think, interesting aspect is that we can now compare a control population with the Kleefstra Syndrome patients. And here you have an example of 3 healthy controls on the left and 3 Kleefstra patients on the right. And without having to understand anything of the complexity of this kind of experiments, I think if you look at the left side, you see very clear similarity between all those controls. You see that there is the graphs, let's say, look very similar. However, if you look to the right, you see that the graphs look quite different. And mainly, you see differences in the boxes that I've highlighted. You see that this is a very long black box. So there are moments of very high activity, and then there are moments of very low activity. And that's very different than what we see in controls. Now, we can do this with very simple cultures with very simple contribution of nerve cells, but we can also make the whole preparation much more complex. And one very important idea in neurobiology is that within our brain there needs to be a balance between excitation and inhibition. And without going to detail, I think you can probably imagine that this is important and that you don't have too much excitation but not too much inhibition, and this needs to be in balance. And so we can also measure this using exactly the same type of approaches. And then we make the whole neurons a bit more complex. We don't generate just one type of neurons, but we generate multiple types of neurons. And then we can do exactly the same exercise. And then again, we can look at this very complicated plots. And what we see now is that by doing those type of experiments, we can actually really pinpoint which cell types in the brain are actually contributing to this different type of communication. And we have started until now with very simple, with very few cell types, but we are increasing the complexity. And of course, as we are increasing the complexity, the whole research becomes more complex, but we also are getting then more to the real situation of a human brain. Now, to summarize a lot of work, I use this slide. In basically on the left, one can say that in healthy controls, there is a certain balance between excitation and inhibition. And what we found in Kleefstra patient cells, so in the dish, I have to really emphasize this, that we see that there is more, it leans more to excitation. There's more excitation. And we think that it is because the inhibition is somehow not working very well. Now, what can we do with this? As I mentioned, we are measuring this really in a dish, and we just started a really exciting project together also with Chitske, where we are now trying to correlate what we find in a dish to what we actually can also measure in the real patients, because this is really important. And how can we compare this? Well, you might all have heard of EEG measurements. Those are actually ways to measure also activity of the brain. You can put this kind of cap on, and then you can look at signals that are very similar of the signals that we can measure also in a dish. And so this project will really aim at measuring both the activity in the dish and the activity of that same patient, so the real activity. And so we try to compare those. And the whole project is we really call it the multi-level strategy. And I think it's really, really important because we don't want to measure only something in a dish and actually not knowing what we are measuring. So we again, from the same Kleefstra patient, we are getting blood samples of fibroblasts. We are generating those cultures, and then we are measuring this excitation-inhibition balance at different levels. And so what is really important is to be able to compare that with real measurements of the patients, of their brain. And why are we doing this? Well, once we have measured this excitation-inhibition balance, so the phenotype, we can now try to, of course, rescue this. And what's interesting, there are actually quite some medications on the market that are not being used for Kleefstra syndrome that can impinge on this excitation-inhibition balance. And so with this strategy, we aim at least to get to uncovering at least certain potential routes for therapy. What does it mean also? I think that this means that I want to end with this so that we can use now stem cell-based experiments to assess individual brain cells from individuals. And this is really essential because we know that there is quite some individual variation at the clinical level. And so we should not assume that even though patients have the same type of mutation, that the functioning of their misfunctioning of their nerve cells is similar across all the individuals. And so stem cells really allow a strategy to now test that at the individual level. And obviously this kind of approach can also be used for trying to predict the potential of new medication, which of course would then need subsequent validation in more complex model systems. With this, I would like to end and thank, of course, all the people in my group that have worked on this and contributed, to people in Nijmegen, and of course the whole team of iDefine that has made this Congress possible. Thank you very much.
Thank you, Dr. Kasri. And then there's a couple of questions in the Q&A that we wanted to cover, but I wanted to also kind of extrapolate on a comment I made earlier about a lot of these drugs are just being for treating the symptoms around KS, and that's not to minimize the opportunities that exist that you kind of just referenced, that there are opportunities for drug repurposing currently out there. And Dr. Kasri, I know that we have discussed before about how in some of your studies, the electrophysiology readouts have had a condition like Kabuki kind of overlay with what's going on with Kleefstra in kind of neuronal activity, which is really interesting. And so perhaps that's a good segue into a question here about what are the medications on the market now that might help treat the excitation?
Yeah, we have tested in this system. So we have tested certain antagonists for very specific channels that are FDA approved, and I think it's still early days because what we, as I mentioned, so we can test this in this kind of culture system, but what you always want to do is really go in a more complex system, and so we are doing this at this moment. So but there are actually quite some compounds or medications available that work on all this excitation inhibition balance, but one really needs to find, there is not just a compound that either increases or decreases excitation. So it's much more specific than that. So one needs to really pinpoint it very well and be very careful for that. So and that requires a lot of research and time. And so we now have some idea of what kind of molecular target we should go after, and that's what we are investigating. So I can't tell you readily okay, this drug will do it. I wish I could, but I think the strategy to get there is laid out, and I think that's important.
Yeah, I think the Brain Model Consortium, what's being conducted there is going to be incredibly important. It's not going to be a study just to kind of level set expectations with the community. It's not going to be a study that happens overnight. It's going to take some time, but there is some really important work being done there.
Yeah, I think it's say that we have scheduled 6 years to do this. So just to give you, and that's a reality. We have to get the cohorts, we have to make the stem cells, we have to record the EEGs from those patients, we have to do all the work in dish, and then try, repurpose drugs to see whether we can, rescue certain, and then if that's possible, we might go to certain end of one studies. But yeah, this will take time, absolutely.
Yeah, and I think there was some commentary in the chat around collecting biosamples, and just to kind of reiterate or add in extra context that those biosamples are what is utilized to help create these iPSC lines. And so I don't know that we could have too many. We want to see, with KS having such a variance, it's important to be able to kind of view the full spectrum, so that when we get to the point of screening a therapeutic, that we can see how it interacts across the spectrum. One other quick question for you, Dr. Kasri, from my buddy Haluk. He asked, will these drugs need to be delivered directly to the brain? And if that's the case, how will they be delivered?
Yeah, this is really a very good question. So, because when we add something into dish, that then we don't have the blood-brain barrier, so we don't have this barrier that prevents drugs to get in. So that's very easy. Then we always have to look at the specific drug, whether it's known that it crosses this barrier. If it crosses this barrier, then we are in luck, let's say. Then there's no, it will reach the brain. If not, there are either chemical changes that need to be made, or there are also ways to get it directly into the brain, which is of course more difficult and more cumbersome. But basically, it will have to be evaluated at the individual level. And I think, yeah.
Great. And then I just saw a question pop in about what year is the study in, and if that's in reference to the brain model study.
Yeah,
this just launched. So the team just received funding, and it's around 8 specific genes, but Kleefstra is certainly one of those, EHMT1.
Yeah, and there is a website, brainmodel.com. We will update there or the progress of so feel free to follow it there, but also of course via iDefine we'll keep you up to date.
Great. Thank you, Dr. Kasri.